Okay, so I want to go over a couple of announcements and then we're going to jump into the course content today with 2 feet. Okay, so I'm going to get very close to finishing the respiratory system today, but it's not quite going to happen. I'm probably going to have about 10 more slides to finish on Monday, and then we're going to jump into the urinary system. Now, I want you to watch 2 screencasts before Monday. It's basically covering introduction to the urinary system. It's about 15 minutes long, and then there's the nephron, which is the functional unit of the kidney. It's what actually produces urine. So if you can watch those screencasts and also complete the self-tests that follow, and then any that you get wrong, go back and look at those slides and see if you can figure out, hey, why did I get that answer wrong? Okay, so don't just look at the screencast and then say, okay, I'm done. Do the self-tests and make sure that you have achieved those learning objectives. Also, this morning, I emailed you the same information, and I also emailed you an image of the nephron. When we start the urinary system, I'm going to go right into how the nephrons make urine. And for you to be in a good position to receive that information, you need to have a basic understanding of the anatomy of the nephron. So if you could use that unlabeled nephron I sent you and label the parts, that'll put you in a good position to start with me on the production of urine, or how the urinary system, nephron specifically, makes urine on Monday. So I know it's a little work I'm asking you to do over the weekend, but it's necessary for us not to have to rush through the urinary system, particularly toward the end. Next week is our last week of class. Can you believe it? It is our last week of class. Okay, just want to remind you of a few things. Your pre-lab on the respiratory system anatomy is due tonight at midnight. If you haven't done that, your lab quiz on the digestive system anatomy from the previous lab is also due tonight at midnight. Your take-home exam is due tonight at midnight, and hopefully you haven't waited to the last minute to do all of these, right? But it is due at midnight. Now, one of the things I suggested that you do with respect to the take-home exam is to share your answers with other students and talk about those answers. If you haven't had a chance to do that, I suggest that you reach out to other students in class. Send them an email and say, hey, I finished the exam. If you finished the exam, let's get on some type of video conference or whatever, and let's compare answers. That is an excellent way to make sure that you get a very good grade on the take-home exam. Your take-home exam, the score should be high 80s or better. So if you haven't done that, if you hadn't had the opportunity to do that, reach out to other students in class. I showed a couple of students how you can very easily email everyone in the class to reach out to them. So I suggest that you do that. Okay. You also have a lecture quiz on the digestive system that's currently open, but it is not due until Monday at midnight. So that's a sort of a run through of everything that is either due or that you should be working on. Okay, so let's now go back to our discussion on the respiratory system. We had just covered pulmonary ventilation, which you know as breathing, and I had told you it's a completely mechanical process, and it depends on volume changes in the thoracic cavity, which then leads to changes in pressure, and then air simply moves from high pressure to low pressure. When we inhale or doing inspiration, the diaphragm falls, the ribs are elevated, the sternum moves anteriorly, so there's an increase in volume. With that increase in volume comes a decrease in pressure, and so air just simply moves from high pressure outside the body to low pressure inside the thoracic cavity, and we inhale. It's an active process as muscles are contracting. In expiration, the opposite occurs. The diaphragm relaxes, the external intercostal muscles relax. The sternum moves posteriorly. The ribs are depressed. And so there's an increase in, there's a decrease in volume, causing an increase in pressure. And again, air just moves from high pressure to low pressure, and air then moves from the thoracic cavity to the outside world. Normal breathing, expiration or exhalation, is a passive process. All the muscles simply relax during normal quiet breathing. If you're doing a forced exhalation, like if you are, if you're running and you're breathing heavy, or if you're trying to blow up a balloon, then muscles like the internal intercostals will contract to force that air out of the lungs. But with quiet breathing, normally expiration is a simple passive process. So going back to this little contraption that I stole from the internet, it basically shows that inhalation is caused by a increase in volume and decrease in pressure, and exhalation, the opposite, a decrease in volume leading to an increase in pressure in the thoracic cavity. And again, air just moves from high pressure to low pressure. Now, there is one space that has a pressure that's always negative with respect to the thoracic cavity, and that's the pleural space. So that intrapleural pressure, that's that pressure in that pleural space, should always be less than the pressure in the lungs itself. And that's so the lungs never collapse. If that pressure ever becomes greater than what's in the lungs, even during exhalation, the lungs would collapse. And we see this, what's called a atelectasis. Atelectasis is collapsing of the lung, and that can be caused by a pneumothorax. If you were stabbed, or for whatever reason there was an entry point made between the outside world and that intrapleural space, it would cause that pressure to be greater than the lungs, and you get the collapsing of the lung, called I always struggle with this word, atelectasis. The way this would be, a pneumothorax is treated, is a chest tube is inserted, and they pull negative pressure and then patch up the entry to that, and then the lungs are able to reinflate. Okay, are there any questions before we move on and talk about respiratory volumes and capacities? Any questions at all? Okay, so let's go over the following respiratory volume capacities: tidal volume, inspiratory and expiratory, reserve volume, residual volume, vital capacity, lung capacity, total lung capacity, dead space volume, and functional volume. And then we're going to contrast bronchial and vesicular breathing sounds. Now, as we cover these various volumes and capacities, I want you to, you're going to be provided with numbers associated with them. However, don't worry about memorizing these numbers. You're not going to be responsible for knowing them. I want you to know the terms and what the terms mean, but I don't want you to memorize a specific volume amount. Also understand that these amounts are going to differ depending on a person's height, their gender, their age, as well as their physical condition. All right, so again, you're going to be provided with typical numbers for all of these volumes and capacities, but you don't have to know them. All right, just want to make sure that's clear. What I want you to know is the definition. And the first thing we're going to talk about is tidal volume, or TV. Tidal volume is typically around 500 milliliters of air, and it's basically the amount of air that moves inside and out of your lungs, your respiratory system with each breath. It's represented by this yellow line here. So every time you inhale and exhale normally, like you're doing now, the amount of air that enters and exits your lungs is called tidal volume, and it's roughly 500 milliliters. It's called tidal volume after the tides of the ocean, because they come in and they go out. Inspiratory reserve volume is shown here in sort of the salmon or pinkish color. It's the amount of air that can be forcibly inhaled above tidal volume. So if you inhale maximally, right, as much as you can, that amount above tidal volume is your inspiratory reserve volume. It's the amount that you can bring into your respiratory system above tidal volume. The opposite is the expiratory reserve volume. If you exhale and empty your lungs of as much air as possible, that volume beyond tidal volume that was expired is your expiratory reserve volume. The expiratory reserve volume. It's the amount above and beyond tidal volume that you can empty of your lungs. I want you to notice that even when you exhale maximally, when you exhale for as long as you can, there is always some air in the lungs, and that's called the residual volume. And there are two very important reasons why we always want air in the lungs, even when we're exhaling. Number one, we don't want the lungs to collapse. If the lungs collapse, we have to spend a great deal of energy re-inflating them. Think about when you blow up a balloon. What is the most difficult part of blowing up a balloon? The beginning, right? When you're first putting air into that balloon. If our lungs collapsed every time we exhaled, we'd have to spend much more than the 5% of our metabolic rate on breathing. We'd have to spend a whole lot more. Two, we don't want our lungs emptied of air because blood is always passing through the lungs, even when we expire, or I should say, even when we breathe out, right? When we exhale. So we always want air in the lungs to participate in gas exchange. Okay? So there's always a residual volume under normal circumstances. The lungs never collapse. There's always a residual volume in the lungs. So these are different volumes. These are amounts that can be measured. We can determine what are called capacities by doing calculations. And the vital capacity is the inspiratory reserve volume, the tidal volume, and the expiratory reserve volume added together. So if we add these three together, this is called the vital capacity, and that's basically the maximum amount of air that you can bring into and out of your respiratory system. So if you do a maximum inhalation and then a maximum exhalation and continue blowing out as long as you can, that total amount is the vital capacity, the maximum amount of air that you can basically move through your respiratory system. If we take the vital capacity and we add it to the residual volume, that is your total lung capacity. So I also want to bring to your attention that not all of the air that enters the respiratory system makes it to the lungs. And therefore, not all the air that enters the respiratory system participates in gas exchange. There is air in your nasal cavity, your pharynx, your larynx, your trachea, and in the bronchi. And none of those structures have alveoli. So the air in those parts of the respiratory system are not participating in gas exchange. They're in the conducting zone. So a portion of your tidal volume is what's called dead space volume. It's not participating in gas exchange, and we call that the dead space volume. It's about 150 out of that 500 milliliters. The other portion is in the respiratory zone. It's in the respiratory bronchioles. It's in the alveoli themselves, and that's and that other 350 is called the functional volume. So it's the portion of your tidal volume that actually participates in gas exchange. So I've shared with you all of these different volumes and capacities. And you may be wondering, well, how are these determined? Well, the volumes, again, are actually measured. There's this apparatus called a spirometer. You breathe into it, and it will actually measure these different volumes. And then we can do calculations to determine these different capacities. Now, you may be wondering, well, why do we care about these various volumes and capacities? What do they tell us about the anatomy or the physiology of a patient, for example? Well, that's a very good question. Well, we can use those capacities and volumes to, one, possibly diagnose a patient, to determine how severe a respiratory ailment they may have, or even chart the progress or the decline of a patient's the health of their respiratory system. For example, with pneumonia and emphysema, you will see a reduction in vital capacity, but the cause of that reduction in vital capacity will be different between patients with these two different diseases. If you have pneumonia, the challenge you will have is inhalation. So someone with pneumonia will have a lower vital capacity, but specifically they will have a decrease in their inspiratory reserve volume. So they'll have a decrease in their vital capacity caused mainly by a reduction in how much air they can bring into their lungs. Someone with emphysema will also have a decrease in their vital capacity, but that will be due not so much because of a reduction in their inspiratory reserve volume, but because of the reduction in their expiratory reserve volume. People with emphysema have a problem exhaling. And we can actually, and emphysema is a chronic condition, and so we could over time, by testing a patient's expiratory reserve volume and vital capacity, we can actually chart how quickly they are declining, or if they're not declining at all. So this is just an example of why these respiratory volumes and capacities are important. So I want to talk about respiratory sounds, or the sounds that are made as air enters and exits the respiratory system. And I usually would play some of these in class, but they don't work as well online. So so if you're interested, I would just have you do those on your own. But we've got two different sounds. We've got what's called bronchial sounds and vesicular sounds. Bronchial sounds are the sounds that are heard as air enters the bronchial tubes. Vesicular sounds are the sounds that are heard as air enters and inflates the lungs, the actual alveoli. And they sound very differently. If you're interested in hearing these, I would suggest that you Google them and then click on the links, and you can listen to them. All of these, of course, are heard using a stethoscope. For the bronchial sounds, you'll tend to have that positioned around the sternum, close, not too far from the heart. For vesicular sounds, you tend to be more lateral, probably around the it's difficult to explain without a visual, but that's typically where the clinician will be using the stethoscopes around your ribs, listening for the sound that the air makes as it inflates and deflates your lungs. A couple of sounds that would indicate abnormal conditions would be crackle. It sort of sounds like, you know how you can bubble air through your drink using a straw? That's sort of what crackle sounds like. It typically indicates inflammation. With inflammation, you get fluid in your lungs, and as air passes through, it makes that crackle sound. Not so different from when you're using a straw to get that last bit of soda out of the bottom of your glass. Wheezing is caused by bronchoconstriction, where you have constriction of the bronchioles, and as the air moves through, it makes a sound. It can sound somewhat like sort of like that. That's not the best illustration of that, but it makes sounds similar to that, and that indicates bronchoconstriction. Something you typically, if you've ever been around someone that had an asthma attack or who's prone to asthma, you've heard wheezing. Okay, so let's move now to external respiration. We talked now about the first event of respiration, which was pulmonary ventilation. Let's move to external respiration. But before we do so, I just want to take a brief pause to see if anyone has any questions about pulmonary ventilation, those capacities and volumes, or those breathing sounds. And just so you're aware, no, you will not have a sound that you will have to identify on the exam. Well, I want you to know what vesicular breathing sounds are and what bronchial breathing sounds are, just a definition. Okay, if there aren't any questions, let's move to external respiration. So we're going to describe external respiration, explain what drives the exchange of oxygen and carbon dioxide across the respiratory membrane, identify the location where most oxygen is transported in the blood, and identify the location and form of carbon dioxide transport in the blood. And I'm moving backwards. Okay, here we go. All right, so just a reminder that gas exchange is made according to the laws of diffusion. So in other words, carbon dioxide and oxygen are going to diffuse from high pressure to low pressure, or I should say high partial pressure to low partial pressure. Therefore, it is imperative that the proper gradients of oxygen and carbon dioxide are established and maintained so that oxygen and carbon dioxide move in the right direction and in the appropriate quantities. Okay, so this image here shows you external respiration and internal respiration. So here, external respiration, we have oxygen diffusing into the pulmonary capillaries, carbon dioxide diffusing out, and then that oxygenated blood is then transported by the cardiovascular system to the systemic capillaries. In the systemic capillaries, we have the opposite movement where oxygen diffuses down its partial pressure gradient to the tissues, and carbon dioxide diffuses from the tissues into the capillaries. Okay, these gradients are established by pulmonary ventilation. Pulmonary ventilation ensures that there is always greater oxygen and less carbon dioxide in the alveoli. And this ensures that there is greater oxygen and less carbon dioxide in the systemic capillaries. Okay, so with external respiration, we're talking about gas exchange between the air of the alveoli and the blood of the pulmonary capillaries. Because air in your alveoli is technically still considered outside of the body, we refer to this as external respiration. Blood returning from the tissues is high in carbon dioxide, low in oxygen. So carbon dioxide is unloaded down its partial pressure gradient, and oxygen is loaded down its, the blood is loaded with oxygen moving down its partial pressure gradient. All right. Therefore, deoxygenated blood is entering the pulmonary capillaries, and oxygenated blood, that's blood that is high in oxygen, low in carbon dioxide, is leaving via the pulmonary capillaries in the lungs. All right. So again, what determines the gradient that allows the diffusion of both carbon dioxide and oxygen? Well, it's the pulmonary ventilation. The reason why we breathe, and I ask students this often before I move on. I said, when we're covering the respiratory system, I always ask, why do we breathe? Why do we breathe? And of course, they always answer, because we need oxygen. We need oxygen. Well, how does breathing actually allow our cells to get oxygen? Because that's really what needs the oxygen, right? Our cells. What role does breathing play in making sure that our cells get oxygen? Pulmonary ventilation makes sure that there is always an appropriate gradient between the air in the alveoli and the blood of the pulmonary capillaries, because that's what drives the oxygen entering the blood and the carbon dioxide leaving the blood. The reason why we breathe is to make sure that there is always the appropriate gradient. If you reduce, like right now, if you want to reduce the amount of carbon dioxide in your blood, how could you do that? Hyperventilate. If you hyperventilate, you drive down the carbon dioxide in the alveoli. That makes this gradient greater, and more carbon dioxide is going to come out, and that's going to lower the amount of carbon dioxide in your blood. It's also going to increase your blood pH, which we'll talk about later, right? That's how you can, if you want to drive down carbon dioxide levels in your blood, hyperventilate. What if you want to increase carbon dioxide levels in your blood? We'll do the opposite. Hold your breath. Hold your breath. What happens? Carbon dioxide builds up in the alveoli because you're not refreshing this air. That reduces the gradient between the pulmonary capillaries and the alveoli. Less carbon dioxide leaves. Therefore, carbon dioxide levels in your blood increase. So the reason why we breathe is to maintain the appropriate gradient between the alveoli and the pulmonary capillaries so that we have proper amounts of gas exchange. Okay? All right, let's look specifically at where oxygen and carbon dioxide are transported in the blood. Okay, and I feel like I skipped a slide. Yes, I did. Okay. So let's focus on oxygen first. So here's our alveolus, and here's our pulmonary capillary. Oxygen is going to diffuse from the alveolus into the plasma, and then it's going to move into the red blood cells. In the red blood cells, that oxygen is going to bind with hemoglobin, which is the protein that's filling that red blood cell, and form what's called oxyhemoglobin. That's HbO2. Now, oxygen binds to a specific location on a specific part of the hemoglobin molecule called the heme group. And that heme group contains iron. When it binds to that iron, it makes iron oxide. Another name for iron oxide is, any guesses? Rust. And what color is rust? It's red. And that is why blood has a red color. And that's why oxygenated blood has a really bright red color, because where oxygen is binding to the heme group, it emits a red color. Okay? This is where most of the oxygen is carried in the blood, within red blood cells, bound to hemoglobin. In fact, if you recall, a red blood cell can carry roughly 1 billion molecules of oxygen. Now, is there some oxygen in the plasma? Yes, there is. There is some oxygen dissolved in the plasma, but it's a minor component of the total amount of oxygen in the blood. And so I've sort of indicated that it's a minor component with this little square around oxygen here. So yes, there is some oxygen in the plasma, but most of it is carried in the red blood cell, bound to hemoglobin. What about the carbon dioxide? Well, when carbon dioxide enters the plasma, which is 90% water, if you recall, it combines with that water to form carbonic acid. Soda pop. You bubble carbon dioxide through water, you get carbonic acid. Carbonic acid then dissociates into hydrogen ions and bicarbonate. So most of the carbon dioxide in blood is in the plasma in the form of bicarbonate. Now, a small portion of carbon dioxide binds to hemoglobin also, but it's a very small portion, which again I have indicated with this box. Means yes, there's some there, but it's a small amount relative to the amount that's in the plasma. And I want to emphasize that carbon dioxide binds to another location on the hemoglobin molecule than oxygen. It's not competing with oxygen for the hemoglobin molecule. Again, carbon dioxide is not competing with oxygen for the binding site on the hemoglobin molecule. So this is what's occurring in those pulmonary capillaries during external respiration. After being oxygenated, the cardiovascular system is then going to transport that oxygenated blood to the systemic capillaries where internal respiration is going to occur. So let's look at that now. So we're going to describe gas exchange during internal respiration, explain how carbon dioxide levels affect pH, and I also want to introduce you to the Bohr effect, and that's Bohr, B-O-H-R, not B-O-R-E, which you might find some of my lectures to be. Okay, so now we are in the systemic capillaries. So we've got the body tissues, and we've got the blood of these systemic capillaries here. So the tissues are constantly using oxygen to make ATP, and they're constantly producing carbon dioxide. So oxygen levels are greater in the plasma than they are in the tissues. So that hemoglobin is going to unload oxygen to the tissues. Now, believe it or not, these red blood cells don't give up all of their oxygen. They give up about half of their oxygen. So about 50, so blood leaving the systemic capillaries is about what's called 50% saturated. It contains about 50%, the maximum amount of oxygen that it can hold or carry. Carbon dioxide is loaded from the tissues into the plasma. It combines with water to form carbonic acid, and carbonic acid dissociates into hydrogen and bicarbonate. Now, in the previous slides, I focused on bicarbonate when I was talking about carbon dioxide. I totally neglected to mention that, hey, carbon dioxide and water forms carbonic acid, and that dissociation into hydrogen ions and bicarbonate ions means that, hey, the greater the amount of carbon dioxide, the lower the pH, right? Because carbonic acid is an acid, and when it dissociates, you get hydrogen ions. So the higher the carbon dioxide levels in the blood, the more hydrogen ions you have, or the greater the hydrogen ion concentration, and therefore the lower the pH. So as carbon dioxide levels rise in the blood, pH declines. That means you would expect blood in the veins to be of a lower pH than blood in the arteries, or at least I should say the systemic arteries, of course. Okay. Now, why am I emphasizing that as carbon dioxide levels increase, pH decreases? Well, there is this effect called the Bohr effect. And basically what it does is it helps deliver more oxygen to those tissues that need it the most. So let's say you're exercising and you've got skeletal muscles in your leg that are really active. They're using a lot of oxygen. They're producing a lot of carbon dioxide. That's going to increase the concentration of hydrogen ions in the blood of those capillaries. That's going to decrease the pH. What do we know about the effect of pH on proteins? Well, it can alter the shape of those proteins so that they don't function as well. Well, hemoglobin is a protein. That drop in pH can cause hemoglobin not to bind as well to oxygen and cause it to give up more than that 50% of oxygen that it would normally unload. And so the Bohr effect is allowing increased unloading of oxygen to the tissues that need it the most. The tissues that need it the most produce the most carbon dioxide, which lowers the pH, which then allows the red blood cells to, the hemoglobin specifically, to give up more oxygen. So that's the Bohr effect. It helps, it's a mechanism that allows those tissues of the body that are in most need of oxygen to get that oxygen. So this is just sort of a summary of the relative amounts of oxygen and carbon dioxide in different tissues and blood vessels and spaces. So if you looked at the amount of oxygen and carbon dioxide in the air that we breathe, right? You can see that the carbon dioxide levels are a fraction of the oxygen levels. Then when we bring that air into our lungs, right? It looks something like this. It's more of a 2-to-1 oxygen to carbon dioxide. When blood leaves the when blood leaves the pulmonary capillaries, the lungs, the proportion of oxygen, the ratio of oxygen to carbon dioxide looks very similar to that of the alveoli of the lungs, right? Sort of like the 2-to-1. At the tissues, there's more carbon dioxide than there is oxygen, and so you get diffusion of oxygen down its partial pressure gradient and carbon dioxide down its partial pressure gradient. And when the blood that leaves the tissues, the ratio of oxygen and carbon dioxide looks very much like what the tissues look like, more carbon dioxide than there is oxygen. And that returns to the heart. Heart then pumps it to the lungs, and there we get loading of oxygen, unloading of carbon dioxide, and now we're back to oxygenated blood, where we have a 2-to-1 ratio. Again, what I want to emphasize is that only about half of the blood, of the oxygen that makes it to the capillaries, is actually unloaded, right? So blood that is pumped to the systemic capillaries is 100%, or should be close to 100% saturated. It's only about 50% saturated when it leaves. All right, any questions before we move on? Any questions at all? This would be the time to stop and ask if there's anything that you're not understanding, or any questions. Okay. Michelle, it looks like I missed your question earlier. Let me get to your question at the end of today's session, because it looks like you asked back when we were talking about pulmonary ventilation, and it might be a little confusing for me to answer that question right now, but I will address it when we get to the end of today's session. I apologize for missing that question, and thank you for being understanding. Okay, well, we're going to continue to talk about internal respiration and just pretty much just define a couple of terms: hypoxia, cyanosis, and carbon dioxide poisoning, and talk about its treatment. So hypoxia is inadequate delivery of oxygen to the tissues. It can be due to anemia. It can be due to a pulmonary disease where there is improper oxygenation of the blood. It could be, and this could be due to high altitudes. You could have a blocked artery or circulation, any reason where there could be decreased delivery of oxygen to the tissues. This is often accompanied by cyanosis, where the tissues take on a bluish tone. So hypoxia is inadequate delivery of oxygen to the tissues. Cyanosis is a bluish skin color as a result of hypoxia. A specific type of hypoxia is carbon monoxide poisoning. And I want to stress this is monoxide, one oxygen, as opposed to carbon dioxide. With complete combustion, where you have a fuel that is completely oxidated, you get water, heat, and carbon dioxide. With incomplete combustion, where you don't get complete oxidation, usually because oxygen is limiting, you get the production of carbon monoxide. And you typically have some carbon monoxide being produced whenever you have an open flame. Carbon monoxide, unlike carbon dioxide, binds to hemoglobin, and it binds to the same binding site as oxygen. And it binds at many, many orders of magnitude greater affinity than oxygen, like 50,000 times or some number similar to that. So it outcompetes oxygen for that binding site on hemoglobin. Carbon monoxide is odorless, it is colorless, and our bodies are not designed to detect it. So if you are asleep in the house at night and for some reason the exhaust for your hot water heater or your HVAC is blocked, and that carbon monoxide that normally would be expelled outside of the house backs up into the house, you can literally inhale it. It can displace the oxygen from your red blood cells because you have no mechanism to detect and respond to it. You simply die in your sleep of hypoxia. That is why it's so important that you have a carbon monoxide detector on every level of whatever dwelling you live in. By law, landlords have to provide carbon monoxide detectors for your homes if you are renting. If you own your own home, you should have one for every level. It can save your life for simply 30, or they're probably even cheaper now. And also make sure you test them every month. Okay, every now and then you hear of someone who dies from carbon monoxide poisoning, and it's completely preventable just having a carbon monoxide detector. And unlike a smoke detector, like smoke detector goes off, right, and then you can stay in the house if you know there's no fire. Carbon monoxide detector goes off, you leave immediately, right, and you call the fire department. Okay, so that ends our discussion on the 4 events of respiration: pulmonary ventilation, internal and external respiration, and gas transport. I now want to move to how pulmonary ventilation is controlled, or control of breathing. So we're going to list the muscles that are responsible for breathing, or at least the main muscles, not all. Identify the parts of the brain that control breathing, and the nerves responsible for transmitting motor output for control of breathing. And then I want to define eupnea, hyperpnea, and hyperventilation. And that's going to be all for today. Okay. We'll talk about conscious control of breathing, emotional control of breathing, and control of breathing by chemical factors, principally carbon dioxide and pH. All right, so breathing is under control of the nervous system. That's probably not major news to you. The main muscles, not the only muscles, but the main muscles that are involved are the diaphragm and the intercostal muscles. The pons and the medulla oblongata control breathing. They are your they contain the respiratory centers. The medulla oblongata have these self-excitatory pacemaker cells that spontaneously generate nerve impulses, and they drive your normal breathing rate. The pons sort of help smooth out the transition from inspiration to expiration, right? You don't have to know those subtleties, just as long as you know pons and medulla contain the respiratory centers. Nerve impulses are transported to the diaphragm via the phrenic nerve and to the intercostal muscles via the intercostal nerves. You can overdose in sleeping pills or morphine or alcohol and cause respiratory arrest as you suppress the normal action potentials that are sent by the medulla oblongata. So if you ever heard of people who overdose on sleeping pills, morphine, alcohol, it's because they basically depress the activity of the medulla to such a level that breathing is below levels sufficient to maintain adequate oxygen to the tissues of the body. Couple of terms here. Eupnea is normal respiration rate, typically between 12 and 15, right? So that's the sort of the normal, the rate of breathing that probably you have right now. Hyperpnea is when there is an increase in respiration rate due to an increase in oxygen needs, such as occurs with exercise. Your breathing rate increases because you are working your muscles more, they need more ATP, therefore they need more oxygen, right? With hyperpnea, we can move from expiration that is passive to expiration that is active. So during eupnea, expiration should be completely passive as muscles just relax. With hyperpnea, expiration often becomes active as muscles are contracting to decrease the size of the thoracic cavity. Is there conscious control of breathing? Well, of course there is, right? You can hold your breath for various reasons, trying to get rid of hiccups, diving on the water, etc. Your cerebral cortex basically sends impulse to your medulla and pons and says, hey, I want you to take in consideration my wishes when it comes to inhaling and exhaling, right? However, your control of breathing is limited. If you try to hold your breath for too long, your medulla and pons will simply ignore your cerebral cortex and say, I don't care what you say, cerebral cortex, we're breathing because we want to live. So when that little kid that can't get what he wants holds his breath or her breath and starts to turn blue, let them turn blue. Just make sure you clear out anything around them so when they pass out, they don't hit their head or something like that, right? I found this little saying on the internet, which I liked. My kid threatened to hold her breath until I gave her dessert. She's now passed out on the kitchen floor. I don't negotiate with terrorists. I thought that was pretty funny. But seriously, if you're working with a kid that wants to hold their breath to get what they want, don't worry. If they pass, the worst thing that can happen is they pass out, and as soon as they pass out, they'll start breathing again, and they will probably not test you in the future. All right, emotional factors, of course, can affect respiration rate, right? We've all watched a movie where we were like waiting with bated breath, or we gasp because we're surprised, or you may even have had some anxiety where you were hyperventilating. So emotions can affect breathing as well. Okay, I am at my end, and I and I really want to hit carbon dioxide and pH and its effect on regulation of breathing very well. So I'll stop here, and we'll start up from here on Monday, and we'll cover regulation of breathing per carbon dioxide, and then we'll talk about some different respiratory disorders, the developmental aspects of the respiratory system, and then we will start the urinary system. So please make sure that you look at those two screencasts and you study the content of those screencasts so you'll be ready to transition in to the urinary system on Monday. Again, I want to remind you that you have your take-home exam due tonight at midnight, your lab quiz due tonight at midnight, your pre-lab for your last lab, it's the last lab of the semester, due tonight before midnight. Thank you very much and have a great weekend. Michelle, you had some questions. Oh, Eli, I will actually respond to that question on Monday. I actually have a slide that relates directly to that. Okay, Michelle, you had a question.